Now that we've completed the annotation of Rheb-PA and yakuba, we need to verify our proposed gene model and prepare the corresponding sequence files for submission. Our analysis of the CDS by CDS T-BLASTn alignments and the evidence tracks on the genome browser allowed us to precisely define the start and end positions of each of the 5 coding exons of Rheb-PA. To verify that our proposed gene model satisfies the basic biological constraints, that it begins with a start codon, has compatible splice sites, and ends with a stop codon, we will check our gene model coordinates using the Gene Model Checker. Open a new web browser tab and navigate to the Gene Model Checker. In the Project Details section, select D. yakuba for the species name and select the August 2021 genome assembly. Enter NC_052530 as the scaffold name. In the Ortholog Details section, enter Rheb-PA for the ortholog in D. melanogaster. In the Model Details section, select No for errors in consensus sequence. In the Coding Exon Coordinates box, enter a comma-delimited list of coordinates for the 5 CDSs. Commas separate adjacent CDSs. No commas are included within each set of coordinates. Select No for annotated untranslated regions. Select plus for the orientation of gene relative to query sequence, since Rheb-PA is on the positive strand relative to the scaffold. Select complete for completeness of gene model translation. For the stop codon coordinates, click within the text box and the stop codon coordinates will automatically populate. Click on the Verify Gene Model button to run the Gene Model Checker. Once the analysis is complete, the right panel of the Gene Model Checker contains the results. The Checklist tab enumerates the list of criteria that have been checked by the Gene Model Checker. For example, the Gene Model Checker verifies that our proposed gene model begins with a start codon and ends with a stop codon. It also verifies that the splice junctions contain the canonical or standard splice donor, GT, and the acceptor, AG, sites. Some of the items on the checklist have been skipped because they do not apply to a complete gene. For example, CDS1 doesn't have a splice acceptor site, and CDS5 doesn't have a splice donor site. The Gene Model Checker checklist is designed to highlight unusual features in the gene model. Warnings and failures reported by the Gene Model Checker do not necessarily mean that the proposed gene model is incorrect. However, you should provide additional evidence to justify anything unusual, for example, a non-canonical splice donor site. In addition to verifying the basic gene structure, the Gene Model Checker also compares the proposed gene model against the putative D. melanogaster ortholog using a protein alignment and a dot plot. FYI, the dot plot is a visual graphical representation of the protein alignment showing the position of the amino acids for the D. melanogaster protein on the x-axis and the position of the amino acids for the target species protein on the y-axis. Therefore, large gaps, regions with no sequence similarity, and any other anomalies seen in the dot plot can be located within the protein alignment. Click on the Dot Plot tab to examine the dot plot between the D. melanogaster protein on the x-axis and the protein sequence for the submitted model in D. yakuba on the y-axis. The alternating color boxes in the dot plot correspond to the different CDSs in the two sequences. Dots in the dot plot correspond to regions of similarity between the D. melanogaster protein and the submitted D. yakuba gene model. If the submitted sequence is identical to the D. melanogaster ortholog, then the dot plot will show a straight diagonal line with a slope of 1. Changes in the size of the submitted model compared to the D. melanogaster ortholog will alter the slope of this line. In this case, the dot plot shows that the 5 CDSs of Rheb-PA in D. melanogaster and D. yakuba have similar lengths. Compare the length shown on the x-axis to the length shown on the y-axis for each CDS. However, Within a small region of CDS4, the dot plot did not detect sequence similarity between the submitted model for D. yakuba and the D. melanogaster ortholog. To further investigate the dot plot, we will examine the protein alignment between the two sequences. Click on the View Protein Alignment link above the dot plot. The protein alignment shows the comparison of the D. melanogaster protein against the conceptual translation for the submitted D. yakuba gene model. Like the dot plot, alternating colors correspond to the different CDSs. The protein alignment between the D. melanogaster ortholog and the D. yakuba gene model shows that the 5 CDSs are 97.3% identical. The symbols in the matched line denote the level of similarity. Star indicates conserved amino acids. A colon denotes amino acids with highly similar chemical properties. Hence, the tiny gap in the dot plot within CDS1 can be attributed to similar but not identical amino acids near its center. The protein alignment between the D. melanogaster Rheb-PA and the submitted gene model in D. yakuba shows that the gap within CDS4 in the dot plot can be attributed to the differences in 3 amino acid residues. We can view the submitted gene model within the context of the other evidence tracks in the genome browser. Click on the Checklist tab. Click on the magnifying glass icon next to Number of coding exons matched ortholog. Note, a new window containing the genome browser will appear with our submitted gene model shown in the red Custom Gene Model track. Now we see our entire submitted gene model for Rheb-PA in D. yakuba. Before adding a screenshot of this to your annotation form, select the default tracks for the region and set the following evidence tracks, if available, to pack. Under RNA-seq tracks, at least one transcript prediction track, like transdecoder transcripts, at least one splice site prediction track, like combined splice junctions. Under comparative genomics tracks, a comparative genomics track, for example, Drosophila chainnet. Click on Refresh. This image, along with the final coordinates, dot plot, and protein alignment, should be added to Part 7 of the annotation form. Next, you will learn how to prepare the 3 data files that you will submit with your project.